Good day, Honors Biology students. Welcome to Video Lecture Episode 11. Today we're going to finish Section 2 and start diving into Section 3 of Chapter 11. Now, as we concluded, we talked about environmental influence, in particular sunlight and water, and then concluded with temperature. Now, we just got a few more notes to mention about temperature before we move on, and that is temperature affects the color of the cat's fur, that being the Siamese cat. The genes that code for production of color pigment in a Siamese cat function only under cooler conditions. Hence, we see temperature inhibits pigment production. Alright, now let's move into slide 79 and following, which, as we conclude the section, we'll be talking about twin studies and how it helps in studying inheritance patterns versus environmental effects. Okay. Now, last on our journey in this section, we will talk briefly about twin studies. Identical twins provide another way of studying inheritance patterns. It helps them to separate environmental contributions from genetic contributions. In other words, twins, since we know they get the same genetic information, but if they have an effect on them that's afterwards, in other words, it's not genetics, but something else is causing it, then they can tell right away it's not the genetics because they should have identical genetics, okay? In fact, it even states that in the book. It says identical twins genetically are the same, so if a trait is inherited, both of the twins will have it. It's not like maybe they will have it. Scientists presume that when traits are expressed differently in identical twins, that the environment is strongly influencing this. The percentage of twins who both express a trait is called a concordance rate. Alright, now if you look at slide 82, the last slide of this section, it has an image, figure 11.16, which expresses the concordance rate for different traits and compares fraternal and identical twins, which I think is pretty interesting because it's two different types of twins. Alright, so let's move now into Section 3. Section 3 of Chapter 11 is titled Chromosomes and Human Heredity. Alright, as a quick little review on slide 3, once you see these slides here, we'll just briefly review the parts of the chromosome. You can just look over that briefly. Now let's move into the objectives. We've got 4 objectives for this section. The first objective is distinguish normal karyotypes from those with abnormal numbers of chromosomes. Objective 2, define and describe the roles of telomeres. Objective 3, relate the effect of non-disjunction to Down syndrome and other abnormal chromosome numbers. And Objective 4, assess the benefits and risks of diagnostic fetal testing. So let's look at the main idea before we move into this any further. The main idea for Section 3 is as follows. Chromosomes can be studied using karyotypes. And if you look on slide 6, there's a picture of what a human karyotype is. Alright, so let's go ahead and let's connect this content to life outside of the classroom. Have you ever lost a piece to a board game or a piece to a puzzle? With a piece lost to a board game, you might not be able to play the game without it. With pieces lost to the puzzle, you don't get the whole picture. And then if you look on slides 8 and 9, I just gave some examples of a puzzle as well as one of my wife and I's favorite board games. Let's move on to slide number 10. We will see in this section how missing chromosomes are like missing pieces to a board game, only their effects are far more significant to the organism. Keep in mind, if you're missing a piece to a board game, you can go buy a new board game. It's not the end of the world. But for genetic information, that can radically affect an organism, how they function, their daily living, or even their lifespan, so to speak. Now, when studying genetic material, scientists don't just study the genes alone. They study the whole chromosomes by using images of chromosomes stained during metaphase. Alright? Now let's talk about karyotype studies just a little bit more. The pairs of homologous chromosomes are arranged in decreasing size in a micrograph picture called a karyotype. Now if you look on slide 12, you see a picture of what exactly that I'm exactly talking about here. And this is where we're going to end our discussion today. So just take this in. If you have any questions, leave them in the comments below. But I do hope this has been helpful. Have a nice day. Take care.